Okay, so here we go. We're going to do a quick review. And I want to see if you are remembering from far away and long ago, meaning last Thursday. So let's talk a little bit about what you remember. Who remembers where in the brain addiction predominantly is impacted? Billy?
Medial forebrain bundle.
All right, and I have heard from other students who have already been through this class. They say to me, oh, I know what you're teaching right now because the medial forebrain bundle is all up and down the hall. Lots of you are saying it to other students, etc. And so there's been some laughter about that. So we know that the area in the brain is the medial forebrain bundle. That's also, what's another name for that? That's also the
Old brain.
The old brain predominantly. Another?
The limbic reward system?
Yes, well, that's a fancy way to say it, but absolutely. The limbic reward system. And I really appreciate that you don't say center. It's the limbic circuit, reward circuit, or pathway. It's not an individual point, it's a pathway. And we've been talking about it in terms of a specific word. What happens in that area? That I'm trying to get you to think of addiction not as a willpower issue, but as a disease and an injury. Let's not shout out. Injury, yes, an injury, absolutely. So that the area has either been injured from birth on, or that there has been, due to usage, an injury that has led the person to be unable to control their usage, basically. Okay. And we've talked a lot about neurotransmitters that are involved in all of this. And now what we're doing in the context of today's lecture is really reviewing again specifically the component parts of neurons, and then we're going to move into how neurons talk to each other, and we're going to move into how the whole system alters in terms of the addiction. So this was a picture of two neurons, but let's go to the next slide. This, I said, was a terrible, horrible, no good, very bad slide, and we're actually going to stay right here. So this is, again, another artist's depiction of a cell. And there's words that say action potential. We're going to find out what an action potential is. But we've also talked about how frustrating it can be that different parts of the neuron have different names. So Rich can be Richard or Ricky, or maybe various times in different people's lives they have different nicknames, but we still know it's the same person. The cell is still the same cell, it just has different names. So we've divided the cell into three parts when we were talking last week. And the first part had to do with this top third from there up through here. And we called that part the, oh, that was weak. We called that part the
Cell body.
Still weak. We called that part the cell body. Oh, thank you. Cell body. And inside the cell body, there is a powerful part that keeps the cell functioning. It holds the genetic material. It's called the
nucleus.
And then the branching out from that cell body is dendrites. Oh, that was not together. Ready? Dendrites. Okay, thank you. So that's the three major parts that we're looking at so far. We'll be looking at some other things. Then we have this long piece of tissue called the axon. Axon, great. The axon, who can raise their hand and tell me what the axon does? Yes.
What the electrical signal actually travels from one side to the other down the axon.
The electrical signal travels down the axon, and it travels more efficiently and more effectively when what happens, Bob?
Well, the myelin sheath,
Yes.
it insulates.
It insulates, and it wraps around the axon much in the same way that with an electrical cord, the plastic covering wraps around the wires. And then as we get down to the last third of this cell, we have a bunch of branching off again, and these are called
Terminal buttons.
terminal buttons. Okay, this particular picture says axon terminals. We've said that there's multiple names. I just picked terminal buttons. That's the one I like to say. Okay, now we have this word action potential. Well, when we take it apart, we see the word action, and action means movement generally. There's action happening. So an action potential is when the cell is actually having the electrical charge move down through the axon right into the terminal buttons. So let's move on now. That was a good review. There's an incredible amount of complexity within the brain. We are making it like kindergarten simple. But when you start to, if you can even begin to visualize, which I don't think I can, but if you could begin to visualize what 100 billion neurons connecting with 50,000 other neurons to form 100 trillion connections, that kind of gives us a sense of how complex our brain truly is. And that's, again, hard to even get a sense of. One of the things I'd like you to keep in mind, I don't need you to remember all this in terms of test questions, etc., but 100 billion neurons, I'd like you to be able to remember that. And who knows? You know, we don't, no, to my knowledge, no one's ever counted each separate neuron, but we're having a pretty good guess that there's probably around 100 billion neurons. Okay? So here's a group of penguins saying, wow, that's more neurons than penguins. Okay. So what a horrible, terrible, no good, very bad picture. Again, this picture is despicable. Why would it even show such a picture? Well, obviously for simplicity's sake. So here we have the cell body, we have the nucleus. I don't know if you can see it very well, but there it is. And some branching. What a terrible picture. We have an axon. It doesn't have a myelin sheath on it. It's particularly ridiculous because here is a terminal, one terminal. What a ridiculous slide. Okay, why would I put it? Well, the title says axon hillock, and if Tara will click again, we'll find out. Okay, I stripped away as much as I could, or the artist did, I didn't do this, to really illustrate another part of the cell we're going to learn now called the axon hillock. This is a very, very, very important part of the cell, and it is approximately right here where the cell body connects to the axon. So an axon hillock, what is it? What does it do? Why do we care? The axon hillock has been called a bean counter. And remember how we said that there are certain neurotransmitters that charge you up. You want to get going. And there are certain neurotransmitters that say, chill out, be happy. What's the rush? Calm down. So, just on a very simplistic level, if you're prone to anxiety and being keyed up, you probably don't have enough of the calming neurotransmitters that say, chill out. It's not that big a deal. So what if you didn't get your first test to be a 4.0? So what? Chill out. You can study. You can do more. Your second test will be better. On the other hand, some people are so chilled out that they almost need a fire built under them, right? And so there's this beautiful balance. Well, your brain is trying to figure out when is a message important to pass along. So, for example, if I were to say, and I'm making up something ridiculous here, that today I was going 75 miles an hour versus 70 miles. I don't think anybody would have an urgency to rush out and quick tell someone in the hallway, oh my, Carla went 75 miles instead of 70. Who cares, really? That's not information that's really very valid to point on. But if I were to say the answers to the quiz are hidden under the garbage can, the second one down, pretty much you guys would be sharing that information because that's valuable information. Obviously, if we hear a sound in the hallway, that's the little kids going by, we just tune it out. But if we hear a siren, fire, we pay attention. So we're constantly kind of gauging important information, not important information. And the neuron is doing that as well. And based on the neurotransmitters that are being activated in the neuron, this part of the cell, the axon hillock, is counting it up. So if I were to say, you can have the day off today, but I won't be giving you any help in learning this information, some of you would say, oh, that's really good, let's cancel class, and you'd vote for that. But some of you would say, hey, I think I need her help kind of learning this, and you would vote not to cancel class. So we could take an actual tally, and if I'm the axon hillock and I said, you want to cancel class and learn this on your own, I'll count up the people that say yes, and then I'll count up the people that say no. And if the no's win, then we don't cancel class. If the yeses win, we do. So the same thing with the axon hillock. It's counting up, should I run this electrical charge or not? And sometimes it's getting a message, no, don't run that electrical charge. It doesn't matter. Chill out, be happy. And other times it's saying, heck yeah, we got to get this message passed on. It's the axon hillock that counts up the yeses and nos. So where do these yeses and nos come from? We'll get there. Okay, let's go on. If the axon hillock counts yes, pass this on, then the action potential happens. Go ahead. So the action potential, the axon hillock takes charge of whether an action potential happens or not. It lets you know whether there's going to be an electrical charge happen or not, and that electrical charge is called an action potential. Let's move on. Okay, so how much input or excitation does it take for the axon hillock to get excited? Well, it depends. And it depends on multiple factors, which we're going to get to today. Okay, so another terrible, horrible, no good, very bad picture. And one of the things that's important about this is that it, again, as we saw, it's an enlarged picture of one we saw earlier on. It's enlarged and distorted because they really want the emphasis to go here, which is where your eye is drawn. And so this is a terminal button right here. And one of the things that we talked about last week is that the terminal button is where neurotransmitters are stored. And they're stored in special little sacs that have a special name called what? Vesicles. Vesicles. Good. So this is an artist's depiction of a vesicle. What we talked about last week is when the axon hillock, we didn't use that phrase, but we did say that when the electrical charge does happen, electricity comes down the axon, it interacts in this terminal button by causing these vesicles to go to the membrane of the cell and merge with that membrane and release their neurotransmitters. And so once the neurotransmitters are released, we need to find out what happens to them. We're seeing another word that we haven't talked about yet, and that word is called synapse. Synapse means space. So there's actually space between two neurons. They do not touch. That's an important piece. They do not touch. Okay, let's go to the next slide. Here is a cartoonish drawing of, again, a terminal button right here. And this artist has shown you a kind of visual where you can look into the vesicle and you can see his or her depiction of a neurotransmitter. In this case, the neurotransmitter is designed to look like a W. And lo and behold, miracle of miracles, there is a receptor that, sure enough, is designed for that to comfortably fit in. This is the first time we're talking about receptors. Receptors are made specific to neurotransmitters. So they have different shapes. Neurotransmitters have different shapes. So it's a match made in heaven. Remember when you fell in love and you met your soulmate and you went towards each other and you fit perfectly when you hug? It's heaven, right? Well, let's pretend that the receptor is sitting there waiting for its perfect love, and sure enough, here comes the neurotransmitter to fit into it, and they have heaven. They have bliss. They're hugging. They're actually touching. Okay. Now, just like any hug, it can't last forever. I don't know anybody who's hugged for 24 hours. I suppose it happens, but usually you at least have to separate enough to use the restroom. So these guys have to separate enough too. So after a neurotransmitter meets its receptor, it doesn't stay there indefinitely. It sits there for a period of time and then it releases. and we're going to find out what happens when it releases. But right now, the terminology I want you to know is, this is a terminal button. These are vesicles with neurotransmitters. When the electrical charge comes down, these actually come and merge and open up, and the neurotransmitters go into this space called the synapse. And in the synapse, some of them will find their home in a specifically designed receptor made for that particular neurotransmitter. This is something I would like to have you have on your study guide, a picture of this as well with receptors. Now, where the heck are receptors? I would like somebody who doesn't have to say a word, but they don't mind looking a little foolish. Who would stand up here? Okay, come on up. So, this fine gentleman here is what's known as a neuron, okay? So I'm going to take this off a minute, okay? And I'm going to ask you to visualize with me what we're about to see. So, this neuron is not going to talk. He's just going to let me distort him. This part right here, he has inside of here, we all know, what's inside of his head? A brain, right? If I only had a brain. Okay, so in his cell body, there's a nucleus. So we're going to assume this right here is his cell body. And we're going to assume that the brain, when I point in like this, I'm referring to the nucleus, which is his brain. Now the problem is he doesn't have any dendrites coming out of this cell body. So here's where your imagination comes. See this nice arm? Pretend it's not here. Pretend it's actually coming out of his head. Does anybody watch Fringe? Okay, you can go there if you watch Fringe. Okay, so imagine not only is one of these branchings coming off of his head, but 50,000 perhaps, right? So what's nice about this example, it's unfortunate his arm really is on his shoulder, not in his head. It'd be much more convenient for my purposes if his arm was off of his head. But remember that mythical creature, goddess, Medusa, with the snakes? Okay, think of the same thing. Okay, now why I like doing this is he has receptor sites. And that's where the receptor sites are. They are predominantly on the dendrites. Receptor sites are predominantly on the dendrites. Okay, so we know this is a terminal button. We also know that this then, because it has receptor sites, what is this? A dendrite. It's a dendrite. Yes, it's a dendrite. So the release of the neurotransmitter happens from a terminal button, and they are caught in receptors. Now, allow your imagination to keep going. Imagine he has hands all up and down his arm. So he has receptor sites all over the place. Okay, you with me? Yes, okay. Now, let's do something else. Since you're here, I'm going to have a couple more of you come up in a minute. If this is an axon, what would this be? The axon hillock, absolutely. So here's the cell body. We've got all these dendrites coming off, all these receptor sites, and we have a decision maker right here. And if the decision maker, the axon hillock, says, yeah, this is important information, share it, then an electrical charge will continue all the way down. Would you hold on there and lift your foot, please? What do you suppose this is?
Terminal.
This is a terminal button, okay? So, I don't want you to fall over. Oh, it is easier? Okay. So, but we can't have a dendrite holding up a terminal button. My. Okay, now, our example continues to be helpful because he has this. What do you suppose this is? That's his myelin sheath. Okay. Is there one more person who would be willing to come up and look ridiculous? Thank you. All right. Let me turn this off for just a minute. Okay. So, here we have another fine-looking neuron. So once again, This is the cell body. Nucleus. Nucleus. Cell body, nucleus. Receptors. And hold on. And we have a terminal button. Now, oh, I forgot the helic. Okay, and this is? All right, so neuron 1, could you turn this way, please? And neuron 2. If you wanted to communicate with each other, how are you going to do that? So if you could put your terminal button up, please. And what are you going to do with this? Oh, the worst thing happened. What happened? He touched. He touched.
He touched.
All right. Well, somebody touched. It was horrible. I saw it with my own eyes. Okay. I'm so worried you're going to fall over. Are you sure? All right. So we know they don't touch. Actually, for the purpose of scaring you and shaking you up, it's better that they did. So then I could have this huge reaction, and emotionally you'll remember the information better. So, okay. And we know that this space here is a synapse. Very nice. So we have the axon helic who said, yes, share this. It's important. The charge came down through the axon. It was efficiently done because of...
What's wrong with this neuron?
This is a troubled neuron. Okay. So we have this space. What do we know is going on in this terminal button right now? One person, raise a hand. Yes?
Vesicles are being released and merged within the cell membrane.
Beautifully. If you didn't hear, she said vesicles are merging with the cell membrane and releasing neurotransmitters. So right now, in this space, this synapse, there are neurotransmitters floating across, and they are looking for their home, okay, on the receptors. And it could be anywhere along all of these different dendrites. All right, gentlemen, thank you. Well, I'll have you back in a minute.
Thank you.
Yes. All right. Those are some of the finest looking neurons I've ever seen. Okay. It's going to be crucial that you remember this is, when you see a picture like this, that you're going to know this is a terminal button. How will you know? Because there are vesicles and neurotransmitters, and that's where they're stored, in terminal buttons. And you're also going to know, even though you hardly see any of it, you just see a fraction of it, that this is a dendrite. How do you know that? Because there are receptors. All right, next slide. Here is a picture from your book. And again, in this picture, what's helpful to know is that the artists, fortunately for our purposes, have made the neurotransmitters look different than the last one. The last one were W's. This one is a little blue triangle. So how wonderful it is that we can have a visual that helps us know there's many, many, many neurotransmitters. They all look different. They're all different shapes. We also know that this is a dendrite. How do we know it's a dendrite? Because it has receptors. So every artist is going to depict this a little bit differently. And these receptors are specifically designed to fit the neurotransmitter. So once again, the message arrives. We've learned a term that says when that message is going, it's called an
Action potential.
good action potential. So the action potential is happening. The charge is coming down. The axon helix said, yes, share the information. So these vesicles will merge with this membrane, and it doesn't show it. It hasn't happened yet. But the neurotransmitters will be released into the synapse. Some of them will find a home. Some of them will find true love, will find their soulmate by a specifically designed receptor. Here is another phrase that gets confusing: synaptic gap, synapse. We'll just call it a synapse for our purposes. We also have some terminology that's new here. This is calling it a presynaptic neuron, and this is calling it a postsynaptic neuron. And then we have something called a reuptake port. So we're going to get to those in just a minute. Let's move on to the next picture. Okay, actually let's go back. Could I have my 2 neurons come back up if I could, and can I have a 3rd neuron join? Okay, these are our bravest souls in the classroom right here. They're forever going to be on the videotape that will inspire the rest of the students in our program, okay? And what I'm going to do is have you stand right here. Now, just for the sake of getting it so drummed into our head we'd never forget, this is a cell body inside
Receptors.
They're all over. Lots of receptors, lots of receptors. Axon hillock. Remember? What? That sounded a little weak to me. Let's try. Axon hillock. And this? And once again, terminal buttons. And yes, myelin sheath. I'm awfully happy for myelin sheaths. Okay, so would you 3 try and communicate with each other in a straight line? So Michael, you're up here. Turn around, lift your foot if you would. And you're going to put your dendrites fairly close. Okay? Ah! He did it again. Okay. He did it just to watch me scream like a little girl. Okay. So we know the space here is a synapse. And if, here's the key, here's the trick question. Our good friend right here, neuron number 2, here's the trick question. Is he a presynaptic neuron or a postsynaptic neuron? How many say post? How many say pre? What else do you say? Both. He is both. So, all right. So, at this point, he is, oddly enough, it seems like the top of the neuron is postsynaptic because he's after this synapse. So the part of the neuron where there are neurotransmitter receptors is postsynaptic. But what a versatile guy he is. Down here, he is presynaptic. He is before the synapse.
Oh, before.
Pre is before, post is after. So every neuron is both pre and post. The other good news is that we have designed a circuit here, okay? So this neuron talks to this neuron, which talks to this neuron. So they're sharing the news. But in reality, if we had a room that would work, we would put 50,000 of you talking to each other, and that would be a much better illustration of what's truly going on. But for simplicity, we'll do this. All right, thank you, gentlemen.
Thanks, guys.
All right. So you're all going to get a chance to be in this great video in just a minute. Okay. So, Mark, you want to stand up for just a minute? You don't have to do anything, okay? Just all you have to do is stand there like this. That's all you have to do. Yeah, that's it. That's it, okay? So, Carrie, would you stand up?
Oh, no, I can't.
Okay, Carrie won't. Who would stand up?
I'll stand up.
Okay, yes, good. All right. So, I'm going to put you standing right here. That's all you have to do, not another darn thing. Okay, come with me. So, I am a vesicle, and inside my vesicle is this lovely neurotransmitter, okay? I have many neurotransmitters. Now, something just gave me a charge. Woo! So I'm going to move toward the membrane of my cell, and I'm going to release, I'm merging with the membrane of my cell, and I'm releasing this lovely neurotransmitter into the synapse. Well, not quite yet. Okay, so you're in the synapse a minute, da-dee-da-dee-da, doing your thing, and then all of a sudden, there he is! Oh! So you got, yes, you find your receptor. Now, life being what it is, you're not going to stay there forever, so you merge back off. Now, this is where we are. What is going to happen? Hold on, what is going to happen? Build the suspense, tell the story. Here she is, she's met her true love, she's done a hello, a meet and greet kind of thing. Now she's back in the synapse, and what is on earth going to happen to her? All right, sit down, think of you, both of you, and we'll find out what's going to happen. There are a couple things that can happen to this neurotransmitter. You all know that I'm a nutcase over recycling. Well, I learned it from the neuron, because the neuron likes to recycle too. And in fact, here's again multiple terminology for the same part. Here it's called a reuptake port. Next slide. Here's a better picture. In this case, it's called a reuptake pump. Same piece of the cell, but it has a different name. And this is actually somewhat helpful because it's showing you that it's pumping it back up. So let's review. This is a vesicle with a neurotransmitter that in this picture looks like a rectangle. Okay? So a different kind of neurotransmitter. We happen to know what part of the cell this is because we've seen vesicles with neurotransmitters in it. So it is a button. I only heard two people. It is a terminal button. Very good. It is a terminal button, and we know that, okay? We also know over here it says neurotransmitter receptor designed perfectly for that neurotransmitter. So this is a weak. Together? Dendrite. Okay, it's a dendrite. And we know right here is the synapse. Synapse. Okay. So something has happened. We've seen that these vesicles, it's only showing one, but it has merged and it opened up and it released out the neurotransmitter. The neurotransmitter went into the receptor. The receptors kind of did their thing. Some of them found their home, their true soulmate, their receptor made for them. And some of them fit into that receptor and then left. Then they're back here in the synapse. And some of them were never found true love. They never found their receptor. So they've been hanging out in that synapse just kind of doing their thing. So what can happen? One of the things that I just got through saying is that the cell will recycle. And it actually has a part of the cell that will scoop up these neurotransmitters, bring it back into the terminal button, and repackage it into vesicles. It's a very green, good cell. This is smart. It's smart to reuse the neurotransmitters. So let's look at the next picture. Here's another picture. Again, the last one, go back one please. The last one, the two cells were side by side. We've been used to seeing them top and bottom. The next picture is top and bottom. What part of the cell is this? Only Chris got that. What part of the cell is this? What part is this? Dendrite. Here it even gave you the answer. Dendrite. Okay. These are, and these are, I love that all these different pictures have different designs for the neurotransmitters, and it's really clearly showing you that each receptor is designed for the neurotransmitter. Now, you can't see this very well, but it says transporter molecule. That's a whole new term. What are we calling it? No.
Reuptake.
Reuptake pump, because you can tell this artist depicted it like this, and the neurotransmitters are being scooped back up. Okay, so we've had lots of different pictures. One picture showed it as just a space. One picture showed it as kind of a churning kind of thing, and now this picture is showing it as just almost like a vacuum, kind of scooping it up. Okay. This picture is also showing you, and I'm glad it does, that there's more in the terminal button than just vesicles. There are lots of parts in the cell that we're not even talking about. There are enzymes and all kinds of things in a cell, and so we're just noticing those. We're not going to pay any attention to them, but we know that it's a good representation of the fact that there are many things in a cell. Okay. So now let's just kind of review it all again in your mind. Visualize that cell. Visualize the cell body, the nucleus, the dendrites, the receptors on the dendrites, the axon hillock, the axon, the myelin sheath, the terminal buttons. Inside the terminal buttons, visualize the vesicle, visualize the neurotransmitters in the vesicle, visualize that when the action potential happens, the membrane of the cell becomes receptive to the vesicles, it merges, it becomes one. The neurotransmitters are released into the synapse. When they're in the synapse, some of them are going to find a receptor specifically designed for them. They are going to do something when they sit on that receptor, and then they're going to be released back into the synapse. Some of them will be recycled through a reuptake pump. Could you go with me through all of that? Did you visualize all of that? Are you comfortable knowing that? You need to be, because it's getting more complex here in just a few minutes. So do whatever you have to do in your study. If you have to draw this out multiple times, draw it out multiple times. Think about it, visualize it. I love this picture. Love this picture. Love, love, love this picture. Okay, so here we see an axon. The electrical charges illustrated by lightning. Here we see things going on, and I said there are extra things in a cell besides what we've been talking about, but we can clearly see the cellophane-like bags that are holding neurotransmitters. In this case, they're nice balls. Okay. And we can see that these guys are waiting and hoping to become a receptacle for those neurotransmitters. So these would be receptors. Okay. Nice little happy receptors. Come to me, come to me, come to me. Reminds me of a person in a restaurant saying, come into my restaurant. Here's the menu. Come in, come in. Okay. Now, cool. How cool is this? This picture right here is, oh, I'd hope more of you would have gotten that. A reuptake pump. Okay. This guy is grabbing the neurotransmitters, and you can see he's repackaging them into the vesicle, right? So he is our reuptake pump. But wait, there's more. What's the goofy ads that you see on TV? But wait, there's more. Yes, now you get more. Yes, exactly. Oh my. A Cookie Monster type thing. Here, lurking, sneakily lurking, right in the synapse area. This happens to be an enzyme that destroys some of the neurotransmitters. Aha. So, some of you are vigorous, healthy 20, 30-year-olds. Some of you are tired and worn out with gray hair and a bad back. Okay. So I'm guessing that if I were a neurotransmitter, that enzyme probably would say, let's get rid of this one. She's not very effective. But for some of you, the enzyme would go, try to get you. It's kind of, I always visualize it like those nature shows when the lion is going for the gazelle, and the fast ones make it, and the slow, pokey ones with gray hair don't. So I don't know if that's true, but that's how I visualize it. So now we know, hold on 1 second. Well, now we know two things happen, okay? If you're a neurotransmitter and you're in the synapse, you can do your thing and then be reused, or you can be destroyed by an enzyme. Ha ha, what's up here? Another enzyme. Another enzyme in the cell that itself is going to say, we've got too many of these neurotransmitters floating around up here. So we're back to that word homeostasis. Your neuron itself is working to keep you in balance. If you're not making enough neurotransmitters, it's going to try to charge you up, make more. If you're making too many neurotransmitters, it's going to destroy some of them. So you're constantly in balance. Okay. It's crucial that you remember all of this. There isn't anything I've talked about that I don't expect you to know. You have to know how that entire cell works. You have to know how it communicates with another cell. And you have to know that once the neurotransmitter is released from the terminal, or from the dendrite where the receptors are, it's going to either be reused or destroyed. Okay. Here's the next little piece of information, and it's again going to get more complicated. What happens when this connection happens between a neurotransmitter and a receptor? You can see that little bits of electrical charge happen, okay? Less with this one, more with this one. So this guy has had a connection with that receptor, or with that neurotransmitter, and it's caused a small bit of electrical charge. Depending on what type of neurotransmitter it is and how many of them are finding a receptor is going to determine how much electrical charge is happening. So if I referred to this last week when I said, when you have a lake and a pebble gets thrown in, you get a small wave, but it'll eventually dissipate. But if I have a lake and I've got all 25 adolescents throwing rocks in this small little lake, I'm going to get a bigger wave that's going to go much further and generate more electricity in the case of a receptor and a neurotransmitter. This is where our axon hillock is deciding, is there enough? Is there enough electrical charge that it's going to actually say, yes, go? So sometimes it's the amount and sometimes it's the intensity. Let's go to the next slide. Again, a picture from NIDA, and this is another illustration of everything we've been talking about. Once again, we're top and bottom. This is the, weak. Good. At least if you're speaking, I know you're still awake. Okay, so it's the terminal button, and we can see a beautiful, clear, bubble-like thing that would represent the vesicle. So now we have something interesting. We have little star-like designs on our picture. And what do you suppose they are?
Neurotransmitters.
Excellent. Neurotransmitters. They're unique neurotransmitters. In fact, I'm going to tell you which ones they are in a minute. And then these blue things sticking up from the membrane of the next cell is actually receptors. Oh, you're making my heart sing. Receptors. Okay. And we know that this part of the next cell is what then? Dendrites. It's a dendrite. How do we know? Because there's receptors there. Okay. So we also can clearly see the synapse. The synapse. Okay. So we can see that this is merging. It has clearly done it, although we can't quite see the opening, but let's assume it has. We can assume it has because we can see that there are neurotransmitters in the synapse. Some of them have found a home. Okay. This picture is also helpful for another reason. Did every receptor get filled? No. So in a normal life of a cell, not every receptor gets filled. I always make up, and it's going to depend on a million factors, but maybe 50% of them get filled. Let's just say maybe 50% get filled for the sake of our examples. So that's normal. It's normal that some of them stay empty. And then we have a piece of the cell over here. What do you suppose this is? It's the reuptake pump. And it looks like one of those carnival rides. What do you call it? Hammer. Is that it?
I think it's zipper. It looks like it.
Zipper?
Zipper, it's the zipper.
All right. Well, whatever. It looks like it's going to operate that way, that it's going to pick up and carry the neurotransmitters back up. But wait, there's more. Oh my, what is this? It's labeled cocaine. So what are we picking up right here? Cocaine has a part of its structure that matches close enough the natural neurotransmitter. it matches close enough that indeed it can actually plug up or block the reuptake pump. It's not allowing the dopamine to go onto that reuptake pump and get reabsorbed. So what would happen? What would happen? It would become flooded. It would become flooded here. Oh my, if it becomes flooded, more receptors will get filled just because there's so many in that area, okay? There are so many neurotransmitters building up. The enzyme can only take care of so many. So there's so many building up that it's flooding, and because it's flooding, now we have really every receptor getting filled. What do you think that is? That's a high. That's what we talked about, euphoria, okay? So here is dopamine. We know it's dopamine because it matches cocaine. And cocaine and dopamine have similar structure, similar enough that the cocaine actually can fit into the reuptake pump and cause it to get plugged up. And therefore, it's going to build up dopamine in the synapse, and it's going to cause those receptors, those that were kind of taking a break, to get fired, and all of a sudden, we're operating at a high peak level.
So, when you're feeling that high, is it the actual cocaine, or is it the fact that...
The dopamine has increased.
Okay.
Let's do an illustration here. Okay. This particular picture is in the medial forebrain bundle, and it's working with dopamine and reuptake pumps, okay? What about if it's in a different part of the brain, and now instead of it being cocaine, it is a pharmaceutical drug I'm going to tell you about here in just a minute, and let's assume this isn't dopamine, but serotonin, okay? And let's assume that the cocaine now isn't cocaine, but Prozac. These become SSRIs. Okay, let's think about this a minute. This is now in a different part of the brain, okay? It's in a whole different part, and it's working on a whole different neurotransmitter system, and it's a whole different drug. But the operation of how it works is the same. Prozac is going to block reuptake pumps so that serotonin can build up in the synapse and actually help charge some of those receptors. Now, why would you do that? One of the reasons you might do that is because the person, through the disease of depression, has made less. They have had a crisis in their brain. They're not getting enough serotonin naturally for whatever reason. And so one of the ways to help that person get back to feeling good from depression would be to actually alter this system in a way that would allow, maybe if our example is that 50% of receptors have to get filled for it to be a normal function, let's say somebody in depression is only filling 10%. Okay, so they're not operating well. They're seriously depressed. If you want to help them, what you're going to do is block. One way you can help them is to block the reuptake pump so that the ones that are there would be more effectively used so that you can get up to 50%. Okay, does that make sense? So SSRI means selective serotonin, here's the good news, reuptake inhibitor. So it inhibits the reuptake pump, reuptake inhibitor for serotonin, and a selective type of serotonin. There are multiple ways. I just said there are 8 ways that this system can be affected. I've just shown you one, okay? But you've already, because you're thinking this through, you've already leaped to another one, which is that you could do something, you could intervene in the system to actually create more of the serotonin or the dopamine or the GABA or the whatever. You could have a drug that would impact how many are being made so therefore more would be released. You can also have a drug that would do other kinds of things. You can have a drug that would cause the neurotransmitter to stay on the receptor longer. There's multiple ways to intervene with this system. So I'm going to hold questions here for a minute. And I want to review this one last time before we take a break. We're going to go give your brains a rest, but it's going to get more complicated from here. So the other thing, another way, I bet you've guessed it because you're thinking ahead, another drug could stop the enzymes, couldn't they? So you've already thought that through. So we already know 3 of the 8 ways. We can have a drug that blocks the reuptake pump. We can have a drug that causes more neurotransmitters to be made. We can have a drug that, or many drugs that do all of these things, but a process whereby the reuptake, or excuse me, the enzyme that destroys the neurotransmitters is shut down. So those are 3 ways we've already come with. The reason I said to you this information is so crucial is as you go into each of the drug categories we're going to study, you're going to want to know, well, how is it working? What's going on when I take meth? How does meth vary from cocaine? We'll find out. We're going to learn, how does opiates work? How does alcohol work? What are all these different ways that each drug has a different impact on this brain cell process? And it's pretty interesting because once you get it, you usually tend to remember it. And then as you start to think about both pharmaceutical drugs as well as street drugs, it all starts to make sense.